I would like to welcome our speaker for today. Elisabeth Kugler joins us, and she will tell us about how to quantify a zebrafish. So this is, I think, quite an intriguing title for a presentation. Elisabeth did her undergraduate studies in microbiology at the University of Innsbruck and then a PhD at the University of Sheffield. And during her PhD, she developed image analysis pipelines for quantifying the zebrafish brain vasculature. And she's continuing now this work combining both biological and biomedical imaging as well as image analysis as a research fellow at the University College of London. And we're really looking forward to hearing your talk today, Elisabeth. Please go ahead.
Thank you so much for this really nice introduction, and thank you so much everyone for joining in. I'm really excited to be here. So as today is about bioimaging, I thought I really take this opportunity and take a step back and think about really the time when I myself fell in love with microscopy and data sciences. And really throughout my education, I was fortunate enough to be really able to study many different biological processes across different scales. And that included cells, tissues, and organisms. For example, at the University of Innsbruck, I studied with Elisabeth Kugler the dynamics of mitochondria. With Pierre Ansted, I studied the role of a certain protein in cellulogenesis in an organ called the Cooper's vesicle, which is required for left-right axis formation. With Darren Gilmore, co-supervised by Dirk Meyer at EMBL, I was studying the role of FGF signaling in collective cell migration using the lateral line as a model. And with Thorsten Schwerte, I studied the role of ethanol in inducing steatosis and steatohepatitis. And so really all these different processes and studies really allowed me to gain this appreciation of really all the experimental opportunities that we do have, be it fluorophores, microscopes, or really just this ability to study processes in 3D in living animals. And it was really just at my PhD level that all of this kind of multiplied by 100 times because I had this absolutely outstanding opportunity to work with light sheet microscopy. And so at the University of Sheffield with Tim Chico and Paul Armitage, I conducted my PhD where we had a size Z1 light sheet, and we were interested into understanding the brain vascular development in zebrafish. So just very briefly, for those of you who haven't seen a fish before, this is a larva at 4 days old. So you look at the back of the head basically. This is the left eye and the right eye. The brain would be sitting here, and here's the trunk, and that's the respective vasculature. So here would be the brain, and we'll be focusing on this region for now. And this is the side view where you look at, again, the eye, the brain. This is the yolk supplying the nutrients to the fish, and this is the trunk. And this is again the vasculature. And for some of the talk, we will be talking about these vessels here, which are the vessels that migrate and form between the muscles or the somites within the fish. But for now, I kind of want to come back to the brain and really say, even though the data are being shown as 2D maximum intensity projections, they're actually three-dimensional. And so I first want to show you this 3D rendering of the brain vasculature and really kind of highlight the sheer size of this field of view. Because if you look from the left eye to the right eye, this size is 600 micrometer, and the depth is actually 250 to 300 micrometer. So it's really a very, very large field of view that we're interested in when we capture the whole brain vasculature in zebrafish. And so I spend a lot of time on this microscope. And so really a lot of different aspects were things that I needed to consider and really spent a lot of time on kind of investigating. For example, what is like XY resolution, Z resolution, colors, time, angles, cell health, and animal health, and how all of these really are together really important to really push the image quality and image properties to the standard that we needed, and having it in such a reproducible manner that we can really study various different aspects of brain vascular development. And once I understood the acquisition, I then said, okay, now what we want to do is actually quantify the vasculature automatically and produce this software. But actually, it is very, very challenging if you don't know at the very start if these fish would be comparable at all whatsoever. And so we started off with, again, some data exploration, which was done manually. And so one thing is, for example, measuring the brain width and seeing how this is variable within age groups and how this changes, for example, over time. And so what we did is measure from this left vessel to this right vessel, so it's the same one as shown here, which is basically surrounding the brain. And so we can use this as an approximation of the width of the brain. And so we measured this width from 2 to 5 days post-fertilization. And what you see is actually that you have a very low variability within the age groups, and actually the changes of size are very comparable between the different fish. And this, together with many other measurements of different vessels or length or angles, really showed us that when we do quantifications, we suggest that actually within the age groups there's a lot of comparability. So we would be able to compare one fish to another as long as they are age matched. And so this was really reassuring. But in addition to all of this really telling us something about biological variability, this also told us a lot about really how we do the imaging and how we can improve the imaging even further. And I do mean by this taking these measurements and improving the field of view, region of interest, as well as resolution. So if we, for example, go back to this example from the width from the left brain to the right brain, we know that this is about 600 micrometer at this age, and so we would set the image width to about 640. So we would allow for some wiggle room, but actually we minimize the empty voxels within the images, and so therefore we reduce the data load down the line for any data acquisition. And similarly, when we look at the side view of the fish, we set a certain vessel as a cutoff point. So we would have a ventral vessel called the basilar artery as a cutoff to go into the brain. And similarly for the dorsal part, we would have this vessel here to always have a standardized measure of the region of interest. And similarly, like I said, this also informs us on the resolution, because obviously when you then look at the vessels a little bit more in detail, you know that some of them are luminized and others are unluminized. So they have different intensity distributions and properties in terms of computational analysis. But also you have very small vessels and very large vessels. And so these are just some of the considerations that you need to take into account. And all of this, like I said, really informs us on the field of view, the region of interest, and the resolution. But this is really just to highlight that at the start, I always spend a lot of time on kind of trying to find out what are the data properties, what do we need to improve, and how does all of this really lead into data quantification later on. But all of this is three-dimensional, and like I said, line sheet microscopy is amazing because also it opens up this dimensionality to time. And in vascular development, there are many different processes that are hugely interesting. One of them is angiogenesis. Angiogenesis is a process where vessels form from already existing vessels, and you can see this here, where so-called central arteries, didn't like my video, apologies. Right, can you see that again?
Yes, we can.
Sorry about that. I had a feeling that the videos would be a bit much. So, never mind, we're back. So, central arteries in the brain are the ones that are formed by angiogenesis, and you can see those here. So, these are forming from existing vessels, and this is happening over a few hours. We can also study other processes, such as anastomosis, which is similar to angiogenesis, but in this case, what happens is actually that two vessels which are already formed merge together to form a new vessel. And in this case, we can see this here, and this is happening in these trunk vessels, which I've mentioned at the start, which are growing between the muscles of the trunk, and they migrate backwards to the very top of the fish, and once they hit this posterior part, they start to migrate to the left and the right, and then the very tip cells try and find each other, as you can see here, and form a new vessel called the DLAV. We can also study, obviously, processes that are happening in shorter time frames, such as, for example, luminization, which is a process where a vessel which was previously unperfused, so it had no blood in it, then has blood in it after this process. And this is shown here in an 8-minute time lapse where you see this kind of bubble-like structure, which is called inverse membrane blebbing. And so this process happens only over a few minutes, and you actually see it in both of the vessels happening. And so it's a beautiful process that you can observe how a vessel actually gets perfused. But all of these studies really were kind of very observational, and so this is important to understand that we spend a lot of time on just looking at these data before we even do any quantifications. But with all of this, what we were able to do in the TICO lab is actually study and prove two new processes that haven't been described in zebrafish before. One of them is neurovascular coupling in zebrafish that has been described by Karishma Chabria in 2018. Which studies the brain neuron calcium imaging together with this disposition of blood cells, so blood flow, upon visual stimulation. And the other process that we were able to discover and describe is actually a membrane behavior that hasn't been shown before, which is called Kugeln. So Kugeln is the German word for sphere. And as you can appreciate, these cells are very ball-like or sphere-like, and they actually sit on top of these vessels, and this is why they're called Kugeln. And actually, these structures are highly dynamic. So if you look at this time-lapse here, you can see two of them. And what you see is that they protrude from the parent vessel and form this ball-like structure. And then some of them change their shape, others maintain their shape and then retract back into the vessel. So they do something. And so in 2018, in EMBL report, we were able to describe these structures and a lot about their cellular architecture in terms of actin and nuclei, etc. But to this day, we don't really know that function. And so this is really still ongoing work in the Chico lab in Sheffield. But all of this is really just kind of to highlight that microscopy is obviously amazing and you can produce many, many data. But now the question is, how do you go from all of these data to something quantifiable? And this is where I sit as kind of biologist/data analyst, trying to produce this software to really go from data to something quantifiable and meaningful for biologists. And here again, you need to consider various different things, such as your image size. So this is where your field of view and your region of interest, as well as resolution, comes in. You need to think about additional dimensions that will add to your data load in terms of channels or time points. And obviously experimental size, such as treatment groups and experimental repeats, is obviously very, very important because it very quickly can add up to hundreds of images. And you need to think about all of this at the very start before you even do anything. And so then when you look at the other side, from kind of the data analysis point of view, if I produce data analysis software, besides the fact that it should work and really be reproducible, it should also have these kind of three aspects. The first one is, for me, usability. If I spend years on producing something, I don't want to be the only person using it, but I want to share it as widely as possible. I want other people just, take it and go away with it. And that also goes with adaptability. So even if I might really optimize it for the zebrafish brain vasculature, I want it to be really dynamic enough to be adaptable to other vascular beds or, for example, other specimens. And where possible, I want it to be open source. And I know that's not always possible, that's why there's this little asterisk. But I think it's really important that when we share or think about open science, that we have really research software that is really open source. And so for me, the tool of choice is Fiji, because it has all of these features, but also because as a biologist, I kind of find that Fiji has the lowest kind of barrier to being used by actually end users that I have been working with. And I think that's also a very important consideration, that you really kind of want to reduce the barriers to people just, installing it, open it and use it. And so all of this really then kind of led me being in this position between biology and data analysis, like I said. And so for my PhD, as well as my postdoc, I've been really working on producing data analysis workflow. So I already touched quite a lot on the first one, which is on the zebrafish brain vascular quantification. But for my postdoc, I've been working on GliaMorph, which is to quantify glia morphology. And so for the next part of the talk, I want to talk a little bit more in detail about really how we quantify now the brain vasculature and kind of what does this mean kind of in the larger scale of things. And this workflow is called CVQ for short. So it's zebrafish vasculature quantification. And just to kind of step back again, what's the main question? Again, we want to quantify the brain vasculature. One thing that we're really interested in is obviously understanding how the vasculature develops, especially in early embryonic development, because as you can see, there's a lot of changes happening, a lot of growth and remodeling, and really a lot of expansion and elaboration of the vasculature. But the thing where we especially need data analysis is where we have very subtle changes. And this is particularly the case when we look at drug treatments, such as here, where we look at a control fish, which is untreated, and a fish that has actually been treated with a drug. In this case, it is a anti-cancer drug, which is called VEGF inhibitor. And VEGF is a vascular endothelial growth factor. So it is actually for the vascular growth or the blood vessel growth really important. So if we inhibit it, we want to reduce the vasculature really very directly. And this is actually, in fact, being used as an anti-cancer treatment. But just by looking at these two images, I, as a human, can't tell if there is a difference between those two or not. Maybe you can, I can't. But this is really where you need computational analysis for these very subtle differences, where it really does make a difference if there is an impact on the vasculature or not. And so for CVQ, we had the aim to really produce various different outrates, such as the volume or the branching points or the complexity of your vasculature. And so we have three main points that are required now to produce CVQ. The first one is image segmentation, then registration and quantification. And lastly, we'll very briefly talk about application, coming back to this initial example of this VEGF inhibitor. But first I want to talk about segmentation. Segmentation is a step where you extract your vasculature from your data, and it is actually the most crucial step. So you binarize your data going from a grayscale image to something that's 1 and 0. But actually it is so important that I've spent a lot of time on this, and I mean a lot of time in my PhD, and so there's obviously a lot of outputs and kind of protocols, etc. that go into this, and I could talk just hours about this segmentation. But for today, I really kind of want to just assure you we have spent a lot of time on this, and we have made sure that it is very accurate, sensitive, as well as robust. And so I don't want to talk about how we did it, but now just very briefly as kind of the principal steps are: you take an original image, you perform an enhancement or improvement step to just improve your image quality, as you can see here. And then you binarize your data, so you have 1 and 0, so for example, vessels and background. And another step is that we actually draw a region of interest to just focus on this brain vasculature. In our case, we just exclude, for example, the eyes or this very dorsal region, but this is just shown here as an example of drawing the region of interest. But in principle, now that we do have the segmentation, this is really the foundation for any following quantification. And so what we build on this is actually now starting to wanting to examine how similar is one fish to another. But actually when we image them, they are not nicely overlapped, but they're actually at different angles. And so if we would like to compare them, this is actually what we would see. So each colour is a different fish, and they're just overlapped at 2 days post-fertilisation. And so what we would like to do is bring them into one spatial coordinate system, and this is called registration. So we just try and overlap them so we can see the patterns of similarity and variability. And there are various different approaches, and we tested 2 of them. One of them is a manual landmark-based approach where we place different landmarks manually and then they're registered against each other. And the second one is an automatic object-based rigid registration where you just have your segmented images as an input and then it will automatically perform the transformations. But both of them are already implemented into Fiji, and I don't want to talk about the details. If you're interested, all of this is in our most recent publication in development, which is not even typeset fully yet. So this is so fresh, but it's all there, the details, so you can have a look at all of this. But basically, just very briefly, the important thing is that the left one, because it is manual, obviously takes longer because you manually need to go through it and place the landmarks. And also, it is slightly less accurate than the automatic registration. But just to highlight that you can, approach this from different angles, and we actually found both of them in theory to work. Then we take these fish and, like I said, overlap them on top of each other, and then we see something like this. This is the same 6 fish, but now being overlapped on top of each other. And now to study the patterns of similarity and variability, what we do is actually produce an average. And this is what it looks like. So in blue, we see regions of dissimilarity, so they're not very similar, and in yellow, they are actually very similar. And just to really highlight and re-emphasize this a little bit more, I have these 2 examples down here. So we call these population average maps, or PAMs, just to kind of refer to them a bit quicker. But actually what you see here is now a control. So these are 3 days instead of 2 days. And again, it's 6 fish. They're overlapped on top of each other, and they're shown as maximum intensity projections. And the similarity is shown in white, and this similarity is shown, for example, in purple. And if you look at this control, what you see is that the vessels that are the most kind of exterior and the ones that are at the very center are highly similar between those fish. And so we can suggest that within this age group, these vessels are really very similar, and they're not, very variable between them. But actually, if we then look at a treatment group where we have no blood flow, so this is achieved by a certain morpholino approach that we inject basically and stop heartbeat from development, so they never have a heartbeat. What we see is actually that they lose these patterns of similarity, and particularly they lose this in this midbrain region. So where these vessels are really similar, they're lost here within these fish without blood flow. And so this allows you then to very quickly see, okay, there's something going on with patterning, and it's particularly happening within this midbrain region. And so obviously you could then perform more quantifications, overlapping them, etc., etc. But this is just to show that this is an approach to very briefly assess where are patterns of similarity and variability, and how do they differ upon, for example, treatment. But then we also take these data, obviously, and then we perform quantifications, and I will talk about the actual quantifications in a minute a bit, but just as a kind of summary, we quantify 8 different parameters, and we have tested them on 12 different models. In this case, on 6 different drug treatments and 6 morpholinos, and again, all of this is in this development study. But all of this is obviously very abstract, so I thought I'd just bring back this example of this VEGF inhibitor, because showing you is obviously easier than just telling you about it. So I want to come back to this VEGF inhibitor, this inhibitor which inhibits and stops vascular growth and therefore is an anti-cancer treatment. Again, on the top we look at a control, and here we have this treated fish, and this is just an individual. So we then produce these population average maps where we have 6 different samples averaged against each other. And this is again control and your treatment fish, and just by looking in them you already see there isn't really a lot of difference, or at least there's nothing that kind of strikingly obviously jumps into your face saying, oh, this is the vessel that has been changed. And so what we do is actually perform these different quantifications now and letting us inform about whether there are differences or not. So the first thing that we measure is a so-called dice coefficient, which is a measure of similarity between these controls and treatment groups. And what you see is that there isn't actually a significant difference, suggesting that actually the patterning itself is not significantly impaired. But what we do see is actually that, for example, vascular volume or the surface, the branching point, as well as network length and the average radius, all of them are being significantly reduced. And again, this is something that shows us clearly that this is where you need computational analysis. I, as a human, would maybe have spent years just looking at these data and maybe trying to find some differences. But now with this workflow, I plug it in, it probably takes a day or 2 to run a complete data set. And then you get these data and they will objectively tell you where the differences are within your network. And so this is really just one example, but like I said, we have tested it on various different applications, just really to make sure to see really where are the limits, what can we extract and what can't we extract really. But I want to come back now to open science, because again, I'm really kind of wanting people to use the workflows that I do produce. And so CBQ, like I said, has just been published in development, and we have to code online on GitHub with example data, and also there's an I2K course recording from 2020 online, which is a very long session, but if you want to hear more about that, you can really step-by-step go through the individual steps. But I just want to highlight again, it's implemented into Fiji as Fiji macros, but actually I implemented it also as one workflow with a graphic user interface, which is shown here. And this is particularly for users who have no computational background, so they don't really need to do anything other than select some boxes, such as, for example, do you want to perform segmentation? Say yes, say okay, and then select the folder of interest, and then you will iterate over the folder. If someone wants to have any adaptions or has struggles with it, any issues, any problems, please let me know. My contact details are on there. Otherwise, on GitHub, if you want to raise issues, please let me know. Again, I want people to use it, and I want it to be useful for the community. And so this is kind of where we leave this first half of my PhD, and this was all great and fantastic. But then I wanted a change of pace, and then I've changed from my postdoc to the University College London into the lab of Ryan McDonald to study glia cells. And here we produce gliamorph as a workflow. So glia cells, completely different to blood vessels, so now really change of gear from blood vessels to glia cells, and we will change from the brain into the eye. So what are glia cells? Glia cells are known as the caretakers of the nervous system. And they fulfill many different roles, from regulating other cells to metabolism, as well as providing structural support. And to fulfilling all these different roles, they interact very closely with other cells. So glia cells are shown here in green, and they interact with neurons, neural cells, as well as blood vessels and other cells. And so they're very tightly linked to interacting with other cells. And so in Ryan McDonald's lab, we really want to understand their role in the central nervous system, not only in development, but obviously also in health and disease. But studying glia cells in the brain is very complicated, because obviously the brain is encapsulated in the skull and it's billions of cells, and so we need to have something that is slightly more simplistic in its architecture. And so what we do is we take the retina as an approximation, where we also have glia cells. And so the retina has a very stereotypic layering, as you can see here. So they have an apical and basal part. on the apical part, we have photoreceptors, then we have neurons. We have a very specific layer, which is called the inner plexiform layer, where we have synapse terminals interacting with each other, which is basically the neuron highway, because a lot of circuits are really having here their interactions. And then we have a layer where we have the blood vessels within this ganglion cell layer. And if you look at the schematic here, what you see is actually there's one cell type, namely the glia cells, that span really from this apical to the basal part, and thereby also contact all the other cells within the retina. And in zebrafish, we only have one glia cell type in the eye, and this is called the middle glia cells. And so to fulfill all these different roles, they need to have the specific shape that allows them to have all these interactions. And this is what it looks like. So this is now an individual glia cell, and we'll talk about these structures in a minute. But I actually also wanted to show you what it actually does look like when we do the imaging first to give you an orientation. So this is again the fish being embedded on the side. Here is the eye, and we image here at this posterior part to always have again a standardized region of interest. And we have a transgenics that allow us again to visualize the different glia or other retinal cell types in vivo. And actually here would be the lens, and here would be the outer part where the photoreceptor sits. And for the rest of the talk, we basically switch it on its top, and we have the photoreceptors here on the top. But just for reference, this is kind of what it looks like when we look through the microscope. And so back to the actual structure of these glia cells. So they have 5 different subdomains, and each one of them has a very specific function. So 1 and 2 is where they interact with these photoreceptors and certain types of neurons. Number 3 is where their cell bodies sit containing the DNA. Number 4 is where they have very, very fine protrusions that migrate laterally and interact with a lot of these different synapse terminals in this really kind of very highly active and really dynamic layer called the inner plexiform layer. And lastly, they have a structure called the end feed, which is actually ensheathing the blood vessels and therefore allowing import and export of compounds into and outside of the retina. And so this really makes clear that, again, their shape is really needed for their interactions with blood vessels as well as neurons, but also we see really their shape associated with their development and maturation, which is shown here where we look from 2 days to 5 days, and you see over time that we go from this progenitor-like, more rounded and undefined cell really, to something that is highly defined with 5 different subregions. We see really these protrusions increasing not only in kind of their size and complexity, but really also like in the numbers really that we have. And we see a downward migration of nuclei happening. And so again, really their shape is associated with their developmental maturity. But also we see changes of shape actually in aging as well as in disease. And so again, quantifying their shape is really important if we want to understand their functionality. And so for the rest of the talk, we will focus very much on this maturation process, but all quantification approaches are equally applicable again to other studies as well. But now, as you can really appreciate, these cells are very different to the brain of the zebrafish. And so if we look here at the scale bar, this is 10 micrometer in size. So these are very, very small cells and also very small protrusions that we're interested in looking at. So with light tube microscopy, we wouldn't have really the detail and resolution that we need in this case. And so what we do use is Airy scan microscopy. And so just very briefly, in Airy scan microscopy, we have this ability of using the different elements of the Airy scan detector, and by them working together, we can increase the resolution and the signal-to-noise ratio in our data. And all of this allows us to push beyond this diffraction limit of 200 and 500 to something of 120 and 350 nanometer. And so if you look at this example image from SICE, this is a confocal image, and this is an Airy scan image of the very same data. So you can really appreciate this huge difference in image quality that you can achieve, which is exactly what we need if we want to resolve these very fine protrusions, particularly here in this IPL region. But obviously, because in this setup of microscope, you have a unidirectional beam path. So the beam goes within the tissue, and so one thing that you kind of need to keep in mind is that the first few layers have an incredibly high kind of data quality and signal-to-noise ratio, but if you go within the tissue, you very quickly have a loss of information and really image quality. And I would highly recommend everyone to read this study. It's absolutely lovely and beautiful to really describing different microscopes and how these kind of tackle this beam or this tissue to light interaction. So it's really beautiful, but just to keep in mind, because I will come back to this a little bit later. But now that we do have these data, like I said, the sub-diffraction resolution, awesome signal-to-noise ratio, absolutely amazing in their quality. Again, we have this question, how can we now actually describe glia cells? Can we quantify their volume or their surface or their thickness or their number of branching points? And so for my postdoc, I'm now producing this toolkit called GliaMorph. So while the CBQ was one comprehensive workflow, GliaMorph is implemented as individual tools. And this is because obviously every kind of experimental approach or experiment is really having different questions. And so to just make it more versatile and more really widely applicable, in this case we have different tools and all of them can just kind of pick and choose and chosen the way the users kind of need them and want them to be used. But basically you can design your own workflow using your original data, and then you do your processing, and then you should derive to have your output data. But again, as I said, this is completely different data to what I was looking at before. So I kind of started again at the very basic of data understanding, processing, segmentation, quantification, dissemination. So it's the same workflow, but completely different data. And so one thing for me, again, was really important to understand these data because I had never worked with Airy scan data and I'd never worked with glia cells. And so again, there are many different aspects that are really important, but I just want to talk about the first three of them because I thought, again, this is really widely applicable to all the people in the audience today as well. The first one is visualization techniques. And in this case, I mean, how do we visualize really the individual cells? Because the standard for us is to use stable transgenics that show or that have a fluorophore that normally is targeted to the cytosol. And this was really the standard of how we visualize the cells within, or the glia cells in the retina. But actually we had a lot of back and forth and discussing and really looking at also the literature, etc. What can we do to improve this? And our amazing technician, Isabel, in the lab then produced actually a transgenic that looks like this. So it's the same cell, but we now, instead of having the fluorophore in the cytosol, we actually tag it to the membrane. And so just by having this different subcellular location, you really see this huge difference in image quality. And this is just one example of it really is worth your time looking at your data and looking at how can you improve all of this acquisition and go really to the biologist and talk to the biologist at the very start. Similarly, like I said, another thing that's important is signal distribution. And like I said, we have this massive Z-axis signal decay. And this is just showing, again, the same example, really like as before. You look at the XY projection and look at this intensity, and it's very homogeneous. But then if you flip the stack to the side, you see that the first few glia cells have a beautiful signal-to-noise ratio, and then you have this drop off very, very quickly. And so we address this computationally in the processing by using bleach correction. But obviously there are different approaches to doing this, but this is just showing now one example where we have the original and then the bleach correction. And so this is really just to highlight that we're so used to looking at our images from one dimension, or from one direction, particularly when we image them in this direction, that it's really always worth not only to look at them in a 3D view, but also really flipping them to the side and spending time looking at, what are any artifacts, is there anything that we need to address in a computational or maybe experimental approach. And the same goes for acquisition, and I know all of you know this triangle of compromise of speed, sensitivity and resolution, and it's very tempting to go to speed and say, just very quickly acquire the data. But again, it's so important to think about how can you optimize your data acquisition, but not end up in like a data overload, because I think that's also like the other end of the spectrum. And so you need to think about like what are your sampling intervals, particularly in the Z-axis, because again, this is not the direction that we're used to looking at. And so if you look at the X and Y, they look both really, really great. But if you actually look at the stack from the side, you see that this is a 1 micron interval versus 0.19. And you see just visually already there's a huge difference in the image quality, right? But like I said, this is 5 times the data load. So you need to kind of find the sweet spot between what is really essential for your data and what can you kind of get away with, so to speak. But all of this is kind of really just considerations, and I find this really important because again, these are the thoughts that you have with biologists and how to visualize and what do you do on the microscope before you even do any data analysis. And then you take all of this knowledge and let it inform your processing. And I don't want to talk about it in too much detail because I briefly touched on it. We do things such as this axis decay bleach correction. We look at point-to-point function deconvolution, etc., but all of this is in the toolkit. But with this, I want to go again to the segmentation, which is this binarization, which comes after your image pre-processing and image improvement. And again, as before, it's the process of taking grayscale images and producing something that's 1 and 0. So in this case, we have 1 is glia cells and 0 is the background. And again, it is really the foundation for volume quantifications or any other quantification. So you really want to spend time on this and optimize it for your specific data set you need to be. But also this is where we automatically trace the skeleton and center line, so it really has further downstream impacts as well. And so for the rest of the talk, again, we look at 3-dimensional data, but in 2D projections. So for the rest of the talk, what I've done is actually color-coded them by depth. So we look at a 2D image, but actually have color codes based on the Z-axis position, just to make it easier to visualize what we're actually looking at. And so we take these data, and I call this segmentation tool, and then put it into the quantification tool. And this is what actually does all the quantification. So this one will take your segmented data and makes measurements of thickness and skeleton and surface to actually then have an output of measurements such as the volume of the surface or the number of junctions. And I will now talk about a few examples to show you really what all of this means. So the first step was to validate and really test if our approach was working. And so we said, okay, we look at a data set at 3 and 5 days post-fertilization, because already by just visually looking at them, what we would assume is that most of the parameters increase over time, such as, for example, the height or the volume. As we can see here, we have an increase in this elaboration really of these structures. But also, for example, the number of junctions should significantly increase, particularly in this IPL region, because you have this elaboration of these protrusions. And so, indeed, most of the factors increased, but I just show you 3 of them due to time, one of them being surface volume, skeleton length, and the number of junctions. And so this was really reassuring, because this was like, okay, yes, we can extract something meaningful, but can we now push it further? Can we, instead of looking at all the cells within our field of view, just look at individual cells? And we could do this with 2 different approaches. The first one is computationally extracting individual cells, and the second one is to experimentally visualize individual cells. For the moment, we're unfortunately not able to extract the individual cells. We're still working on this, and hopefully, cross fingers, this will work. But for now, we work with experimental data. And so in Zebrafish, what we can do is we take a stable transgenic, shown here in magenta, and actually inject DNA at the 1 cell stage to visualize individual cells later on. And this is what is shown here, showing individual clones within this very same stable transgenic. And so we can image them at different time points, and again, perform different steps of pre-processing and segmentation to extract these different cells at different time points. And so we have done this, shown here, again, from 60 to 96 hours post-fertilization. So this is about 2.5 days to about 4 days. And then now I've broken it up into object-based measurements and skeleton-based measurements. And also to visualize these cells, or these kind of progressions a little bit more, what I've actually done is normalize all the measurements against baseline at 60 hours post-fertilization. So it's all normalized to this initial time point now. And what this shows us, if we look at the object-based measurements, is actually that most of the parameters increase over time, such as retina height or the volume, as we would expect. And the only one that decreases is actually thickness, which is again something that we would expect due to the fact that at the start we have something more rounded and progenitor-like, but over time it starts to really elaborate and we get a lot of these very thin protrusions. And we see a similar process or similar kind of patterns if we look at the skeleton-based measurements, where we look, for example, at the skeleton or the number of junctions, and all of them really very nicely increase very similarly over time. And the only one that decreases over time is average branch length. And this is again something that we would expect biologically due to the fact that we have these very fine protrusions, and obviously they're more and more over time, and so this just decreases your average branch length in terms of measurement. And this is all really very reassuring, because now we can obviously look at a whole field of view, analysing stable transgenics, for example, but we can also take this approach to analyse individual cells and track their individual development. And all of this is really exciting, but still a really work in progress. But with this, we then also said, okay, this is amazing. Can we now look at our data from a very different perspective and actually look at it from a perspective of like image intensity? If we look at a maximum intensity projection, what we see is again these 5 different subdomains. And just by looking at it, what you will appreciate is that, for example, cell bodies are very, very bright, whereas in the IPL, we have a very dim signal. So we said, okay, could we make use of this and actually use this intensity information to plot it from this apical to this basal position and let us inform about these different subdomains? And so in theory, what we thought is that if we take this 3D stack and collapse it down into 1D, that we would see something like this in terms of pattern, and then we can just plot it like a heat map like this with apical to basal, and white/yellow is very bright and purple/black is more dim. And so again, to really study this first, we applied it to this data at 3 and 5 days post-fertilization, and indeed we see this downward migration of nuclei and an increase in height. But it actually becomes even more interesting if we do the very same thing for data that have been segmented or the ones that have been skeletonized. And particularly if we look here in this IPL, what you can now appreciate is really that the amount of skeleton at 5 days really significantly increased, and we can actually see this 2 different bands, which is again in finding with what we do biologically. And again, you could do this for other measurements as well, just doing the top to the bottom plotting approach. But I think this is really interesting just to look at your data from a very, very different perspective. But all of this is very much glia focused, and like I said, GliaMorph is implemented as a modular toolkit. And this means you can take certain steps and just apply them to other data, such as, for example, this zonation tool, which is the one that plots your data from this apical to basal pass. You can apply this for Dappler staining to measure your distributions of your cell nuclei. It's just a different representation. Or you can study, for example, neurons as well as their interactions as shown here, where we look at bipolar cells, which are the ones here in blue, which are distributed in this upper half of the image, and we can study how they relate to cells called the horizontal or amacrine cells, which are these horizontal cells here, and amacrine cells are in this IPL. So we would assume this double peak, and indeed we see this double peak distribution here. And so this is just one example of, like, it's not just for glia cells, it's also for other cells, but obviously we have very much optimized it for this. But also, obviously, cellular interactions is hugely important. And so what I'm currently working on is really trying to understand how glia cells interact with other cells. And as you can imagine, there are 2 regions that I'm particularly interested in. The first one is this subdomain of this IPL. So this is where we have these the synapse terminals of the neurons. And this is where really a lot of interaction and really activity is happening. And so what we're trying to understand is really these different synapse terminals and how they interact with the glia cells. And so this is just a 3D rendering of some of the extracted synapse terminals just rotating around. But the other region that I'm particularly interested in is actually how these end feet interact with the vasculature. As I mentioned at the very start of my talk, or halfway through, the end feet actually touch onto these blood vessels, and this is where this interaction and import and export of compounds is happening. And so this is a really interesting interface, and it's not really known how is it formed, how dynamic is it, and what are the processes that do impact this. And so we're taking double transgenics as shown here, where we have glia cells and blood vessels, and now trying to computationally model this interaction surface and really study how this changes over time. But again, this is very much still work in progress. And so with this, again, I just kind of want to come back to this open science and really re-emphasize GliaMorph is, again, implemented in the Fiji. I'm trying to make it as user-friendly and as applicable as possible, so you can take your original data and you have data analysis outputs at the very end. We kind of, just by a few weeks, are off with putting it actually on bioRxiv, so it's a very shame that we just missed that. But yeah, keep your eyes out in the next kind of 2-3 weeks. Hopefully we will have a preprint as well as protocol online. And at that time, we will also share the code and example data publicly. But if you're already interested and you want to use it already or have a look at it, please get in touch with me or my supervisor, Ryan, because again, we're more than happy to share already and get user feedback. Also, if you have data that you would like to look at, just send us an email, because again, we love to work with other people as well. And so with this, I need to thank a whole village of people, and I don't have time to do all of this, but I really want to thank my supervisors, Paul, Tim, and Ryan, who have been absolutely exceptional in making all of this possible. I also want to thank everyone in the labs for supporting all of this, not just intellectually, but also really providing data and input, especially Isabel Bravo and Juliana and Jermishi have been absolutely exceptional, as well as Alicia Carrington, for producing and supporting with data. And I also want to thank all the collaborators as well as the funders, particularly from the University of Sheffield, as well as the Moorfields Eye Charity, because without them, I wouldn't be sitting here today. And so I want to thank all of you for attending, and I'm really happy to take any questions. Thank you for your time.